Tibor Ganti, born in Hungary in 1933, was a biochemist with an insatiable curiosity about the origin of life. From a young age, Gánti was captivated by the complexity of living organisms. He wondered how life could arise from non-living matter. This fundamental question guided his scientific journey. Tibor Ganti's ideas about the origin of life focus on a simple chemical system called the chemoton, which can be compared to a basic fluidic machine. Imagine a small, self-sustaining system like a collection of interconnected pipes and containers where liquids flow, mix, and react. This system has three main parts: a production line, metabolism, a blueprint copying machine, self-replication, and a storage unit, compartmentalization. Gánti suggested that life started when simple chemical systems could perform all three functions. For instance, metabolism in this fluidic machine is like a series of chemical reactions happening within the pipes, producing energy and building materials necessary for the system to maintain itself. These reactions are similar to how our cells convert food into energy. The blueprint copying machine, self-replication, is like a mechanism within the system that ensures the instructions for making new parts are copied accurately, akin to how DNA copies itself in living organisms. Compartmentalization is similar to having distinct sections within the fluidic machine, where different reactions can occur without interfering with each other, much like a cell membrane keeps all necessary components in one place. In 1971, Tibor Ganti published his seminal work, The Principles of Life, originally in Hungarian. This book marked a turning point in his career and in the field of origin of life research. In The Principles of Life, Gánti laid out his vision for a new kind of biological theory. He argued that existing theories focused too narrowly on specific molecules or processes. Instead, Gánti proposed a holistic approach. He believed that understanding life's origin required considering the system as a whole. Gánti introduced the concept of the chemoton in this book. The chemoton was his theoretical model for the simplest possible living unit. It was a self-sustaining chemical system capable of replication and evolution. Gánti's book received limited attention initially, primarily due to the language barrier. However, its core ideas, particularly the Chemoton model, would eventually reach a wider audience. The Principles of Life laid the groundwork for Gánti's later work, the Chemoton theory, which further developed and refined his groundbreaking ideas. Imagine a tiny, self-contained world, a droplet of life adrift in a primordial sea. This is the essence of Tibor Ganti's Chemoton model. More than just a collection of molecules, the chemoton represents a paradigm shift in our understanding of life's origins. At its core, the chemoton is a theoretical construct, a minimal system possessing the essential characteristics of life. It's a self-replicating, evolving entity, a chemical machine capable of maintaining its structure and function while adapting to its environment. The beauty of the chemoton lies in its simplicity and elegance. It distills the complexity of life into three fundamental processes: metabolism, replication, and membrane growth. These processes are intricately linked, forming a self-regulating cycle that drives the chemoton's existence. Think of a car engine. Fuel is burned, metabolism, the engine drives the wheels, replication, and the car moves forward, allowing it to refuel, membrane growth. Similarly, the chemoton's processes are interconnected, each one essential for the system's survival and perpetuation. The chemoton model isn't just a static picture, it's a dynamic dance of three interconnected processes. First, the metabolic cycle. This is the engine room, the powerhouse of the chemoton, where the magic begins. Here, simple molecules from the environment are transformed into energy and building blocks, essential for the chemoton's activities. Imagine a bustling factory, converting raw materials into usable products. This is how the chemoton operates, tirelessly working to sustain itself. The chemoton's metabolic cycle provides the energy and materials needed for its survival and growth, ensuring it can continue its functions. Next, we have the replication process, which is equally vital. Like a blueprint guiding construction, this process ensures the faithful copying of the chemoton's genetic information, a crucial step for continuity. This information, stored in long molecules, dictates the chemoton's structure and function, acting as a master plan for its operations. Think of a photocopier duplicating an important document. This analogy helps us understand the precision required in genetic replication. The chemoton's replication process ensures the transmission of its genetic legacy to its offspring, preserving its identity through generations. Finally, there's containment, the third pillar of the Chemoton model. The chemoton isn't an open system, it's enclosed within a membrane, a protective barrier that defines its boundaries and maintains its internal environment. This membrane, like a cell wall, regulates the flow of materials in and out of the chemoton, ensuring a balanced internal state. Imagine a growing bubble expanding to accommodate its contents. This is how the chemoton's membrane behaves, adapting to its needs. The chemoton's membrane grows alongside its internal components, ensuring its integrity and stability, a vital aspect for its survival. In summary, the chemoton's triad of metabolism, replication, and containment work in harmony, creating a self-sustaining and evolving system. This intricate interplay of processes is what makes the chemoton a fascinating model for understanding the fundamental principles of life. Tibor Ganti wasn't content with simply proposing a theoretical model. He wanted to ground the chemoton in established scientific principles. He believed that a solid scientific foundation was essential for the model to be taken seriously by the scientific community. He drew upon his deep understanding of chemical kinetics, thermodynamics, and kinetic modeling to build a robust framework for his theory. This comprehensive approach ensured that every aspect of the chemoton was meticulously thought out and scientifically validated. Gánti recognized that life is fundamentally a chemical phenomenon, governed by the laws of chemistry and physics. He saw the chemoton as a bridge between the abstract world of theoretical biology and the concrete principles of physical science. He meticulously analyzed the chemical reactions involved in each of the chemoton's processes. This detailed analysis was crucial for understanding how the system could sustain itself and replicate. He calculated the energy requirements and the rates at which these reactions would occur. By doing so, he could predict the efficiency and feasibility of the chemoton's operations. By applying the principles of thermodynamics, Gánti demonstrated that the chemoton was a thermodynamically feasible system. This meant that the system could theoretically exist and function in the real world. It could maintain a state of dynamic equilibrium, constantly exchanging energy and matter with its surroundings while preserving its internal order. This balance was key to the system's stability and longevity. Gánti went a step further, using kinetic modeling to simulate the behavior of the chemoton over time. These simulations provided insights into the long-term viability of the system. These models allowed him to explore how the system would respond to changes in its environment and to identify the conditions necessary for its stability and growth. By understanding these dynamics, Gánti could refine his model to better mimic the complexities of living organisms. The origin of life is a puzzle with many proposed solutions, each offering a unique perspective on how life might have begun on Earth. The Chemoton model stands as a compelling contender, but it's not alone in the race to explain life's origins. This model suggests a complex system of chemical reactions that could have led to the first living cells. Two other prominent hypotheses, the RNA world and metabolism first, offer alternative perspectives on life's beginnings, each with its own set of supporting evidence and theoretical backing. The RNA world hypothesis posits that RNA, not DNA, was the primary carrier of genetic information in early life. This idea is supported by RNA's unique properties. RNA's ability to store information and catalyze reactions makes it a versatile molecule, potentially capable of driving early life processes. It could have played a dual role in both genetics and catalysis. The metabolism first hypothesis emphasizes the importance of metabolic networks in the emergence of life. It focuses on the chemical processes that could have led to life. It suggests that life arose from self-sustaining chemical cycles that predate the evolution of genetic material. These cycles could have created the conditions necessary for life to develop. The Chemoton model distinguishes itself by integrating aspects of both the RNA world and metabolism-first hypotheses. It proposes a more holistic approach. It acknowledges the importance of both genetic information and metabolic processes, suggesting that they co-evolved in a synergistic manner. This coevolution could have been crucial for the development of early life. While each hypothesis has its strengths and weaknesses, the Chemoton's integrative approach provides a comprehensive framework for understanding life's emergence. By combining elements from both the RNA world and metabolism-first hypotheses, it offers a more complete picture of how life might have started. Tibor Ganti envisioned the Chemoton not merely as an abstract concept, but as a testable hypothesis. He believed that its principles could guide experimental efforts to recreate life in the lab. Since Gánti's time, scientists have made significant strides in synthetic biology, developing techniques to build artificial cells and mimic biological processes. These advances have fueled attempts to experimentally validate the Chemoton model. Researchers have created protocells, simple membrane-bound compartments that exhibit some of the characteristics of living cells. While these protocells are far from the complexity of a chemoton, they represent important steps towards building artificial life. Computational simulations have also played a crucial role in exploring the chemoton's potential. Scientists have developed sophisticated computer models that capture the dynamics of the chemoton's processes. These simulations allow researchers to study the chemoton's behavior under various conditions and to test its robustness. Today, scientists still find Gánti's ideas useful. For example, the production line, metabolism in early life forms, can be compared to simple chemical reactions that naturally occur in the environment, helping the system grow and sustain itself. These reactions are essential for providing energy and building blocks for more complex structures. The blueprint copying machine, self-replication, can be seen in the way DNA copies itself, ensuring that genetic information is passed on during reproduction. The storage unit, compartmentalization, is like the cell membrane that keeps everything organized and contained, allowing the cell to function efficiently and maintain its internal environment. However, Gánti thought that these systems needed to have all three parts right from the beginning, like needing a complete fluidic machine to work. Modern scientists now believe that life might have started even simpler, with just some parts working before the full system evolved. For example, early life forms might have had simple metabolic reactions occurring in the environment without needing a full compartment or a complex blueprint copying system. Over time, these simple systems could have gradually become more complex, eventually leading to the fully functional cells we see today. So, Gánti's model is still a good starting point, but we've learned that life's beginnings might have been even more basic, evolving step by step.